everyone. Welcome to our third session on biostimulants. I am Claudia Costa. I am the Academy Director at Food for Sustainability, and I will be your host for today's Grow webinar. And we can say that's been quite a journey exploring the world of biostimulants, right? And if you remember, we have covered what biostimulants are, how they work, and how they are helping farmers. We also dug into seed microbiota and those fascinating endophytic bacteria, if you remember well. And not only this, but we also delve deeper into formulation effectiveness and also all those exciting companies that are tackling now nutrient acquisition and stress tolerance. And today we're going to take things up a notch. So today we'll be looking at how to keep tabs on microbial inoculants in the soil. We will also explore how plant-based biostimulants are giving farmers a helping hand, and then we'll be diving in how something called Leafamine helps plants deal with water stress. And to guide us through this exciting journey, we have the pleasure to have three amazing speakers all lined up already for you. So we will start with Antonio Ferrante. Antonio is a professor with expertise in plant senescence, post-harvest physiology of horticulture crops, and plant responses to biostimulants. And Professor Ferrante will present on advancing sustainable agriculture, the role of plant-based biostimulants. And his talk is expected to cover how plant-derived biostimulants can enhance crop productivity and resilience, contributing to more sustainable agriculture practices. Then Antonio Ferrante's talk will be followed by Loredana Canfora. She's a molecular biologist. She's also a biotechnologist specializing in soil microbial ecology with extensive research on soil biodiversity, integrated pest management, and bio-inoculant detection methods. Loredana will discuss innovative molecular tools for tracking microbial inoculants in soil and supporting their application. And we are convinced that her talk is likely to focus on cutting-edge technologies for monitoring and assessing the effectiveness of microbial inoculants in agricultural soils, and I'm sure she will be providing insights into their behavior and impact on soil ecosystems. And later on today, we'll also have the pleasure to have Alexandre Kerckhove. Alexandre is a researcher with a strong background in plant biology and biosolutions, focusing on the effects of biostimulants on plant stress responses, particularly in the cases of water deficit conditions. Alexandre will discuss Leafamine, a protein hydrolyzed biostimulant, enhancing lettuce tolerance to water stress. Sure, his presentation will likely showcase his research on his specific biostimulants and how it improves lettuce's ability to cope with water stress, potentially including insights from his innovative Rhizobok systems study. So it's with great thanks that I thank you all for joining this discussion, and I will just very, very briefly, I promise, will take this opportunity to remind you that our Soil and Function Laboratory is advancing soil health through DNA genetic sequencing technology and, of course, expertise in soil microbiome, biochemistry, and agronomy. And of course, our goal is to bring practical applications for farmers' problems, namely pathogens, and as such, benefit farmers, agronomists, and researchers alike. Also, I'm excited to announce our Soil Health summer school. And in this program, we'll dive into soil microbes, plant health, and sustainable agriculture. So join us and learn from leading experts to develop your own projects under their guidance. And so I invite you to register now and shape the future of agriculture altogether. Whether you're a student, a professional, or just an enthusiast, let's take the chance to be at the forefront of soil science. So please, I think Soraya has already shared here in the chat the links. Hope to see you all later this year. And because at least here from my side of the world, it's very gray and it's cold and it's miserable. Let's go to do a warm-up. We do have Slido. We have prepared Slido for you and what we have. I think Soraya is already sharing. You can scan the QR code, or alternatively, you can use your browser. And let's all jump in Slido. Let's take this opportunity to enlarge our knowledge on the topic. Sometimes we don't address some of the questions we have here in such a specific matter. So let's see what the team has prepared for us, if we are all ready. So the first question, do you know who is the primary regulatory framework for biostimulants in the European Union? So regulatory framework for biostimulants. And we have here the options of common agriculture policy, fertilizing product regulation, our famous European Food and Safety Authority, or is it organic farming regulations? And as we all know, conformity assessment is a crucial process used to ensure that the products meet specific regulatory standards and also the requirements before they are placed on the market. And conformity assessment includes testing, inspection, and certification by following the procedures outlined in thank you. It's on the fertilizing products regulation. So there's this body. It's not EFSA. EFSA enters at a different stage, but those are the ones that define the regulatory framework for biostimulants in the EU. So at least now we know. This was news to me also, but it's a good piece of knowledge. And now, as we heard also a lot during our sessions, biostimulants can claim a lot of benefits, but they can only come to the market with one single benefit. So which of the following is a key benefit of biostimulants in modern agriculture? Is it because they reduce pest spread, so they have an effect in terms of pest control, because they increase tolerance to stress factors, because they replace chemical fertilizers, or is it because they guarantee an increase in crop yield? And I think that's very consensual, our audience here. So indeed, you're absolutely right. Biostimulants can help plants better withstand abiotic stresses such as drought, heat, and cold. They indeed promote the production of stress defense compounds and stimulate plant metabolism, making them more resilient to stressful conditions. Thanks. It was indeed one of the main benefits is indeed increased tolerance to stress factors. And we already have our third question. So very famous names here on the screen. So are you aware which organization is leading the global standardization efforts for biostimulants? And is it the WHO, World Health Organization? Is it FAO, Food and Agriculture Organization? Is it the ISO, the International Organization for Standardization? Or is it the World Trade Organization? So FAO indeed has a lot great recommendations in terms of how to move forward, in terms of agriculture and sustainable agriculture, regenerative agriculture. We have a vote. But in this case, for standardization, the body that is looking at ensuring that we have global standards is the International Organization for Standardization. It's ISO, which is an independent, non-governmental international organization with a membership of 164 national standard bodies. And it's through its members that it brings together experts that share knowledge, develop voluntary, consensus-based, market-relevant international standards that supports innovation, of course, try to provide solutions to global challenges. So standardization is indeed ISO, the International Organization for Standardization. Thank you so much. Thanks for having here. It's great to see you taking part, and hope you also learn. So let's move to our speakers that are here eager to share their knowledge. And I will start with Antonio Ferrante. Antonio, please come to the floor. The floor is now yours. Thank you.
Thank you very much. It's a pleasure for me to be here and present our activities and research on plant biostimulants. So today I will talk you about the plant-based biostimulants that can be an products that can help in improving agriculture systems. Since we know that in the future we have to face several constraints and different challenges we have to face, like the global climate change and the increase of world population. That means that we have to pay more attention to our production systems. Of course, we have a change also in terms of consumer request, and so we have to provide foods and products that are high quality. But in the meantime, we have to save the resource and improving the productivity without having environmental impact. So we can work in different levels. For sure, we have to work on crop improvement through genetic techniques, but we have to find in a short period some new tools that can help the production system. And on this view, we can improve agronomic management using biostimulants. What are biostimulants? So we know that they are included in fertilizer regulation. So there is an European regulation, and all countries has to consider this new organization in terms of commercial products. But biostimulants, as the word says, they are focused on the activation, some biological pathway and biochemical pathway that lead the plants to improve yield quality or increase the tolerance against abiotic stress. At least this is what the law wants to define biostimulants. So in terms of to provide products against biotic stress, we have to talk about biological agents. So biostimulants can help in improving sustainability of a cropping system. They can reduce, for example, the nutrients application through fertilizers, can improve the nutrients use efficiency. They can improve the resource utilization, like water, or faced against some abiotic stress like drought, salinity, and high temperature, and so on. And the main compounds that have a biological activity can be humic acids, some carbohydrates, peptides, amino acids, vitamins, and they can be obtained from different raw materials like seaweeds, but also, for example, microorganisms that can produce this compound. They can be applied to the soil or to the plants, and they can improve directly or indirectly the productivity of our cropping systems. This is in line with the European Green Deal wants in terms of reduction of fertilizer, especially here in the point 2 of the The target is to reduce the losses in the cropping system through the 50% from soil and reduce the 20% of fertilizers. This is very important because we can use biostimulants in order to improve nutrient use efficiency, reduce fertilizers. It can be also a benefit for agriculture, organic farming in terms of the utilization biostimulants. We know that biostimulants before the uh, uh, 1009 regulation of the 2019, we had the different compounds that can products that can be classified as biostimulants. And now with the law regulation, the new regulation, some of them are out, but for sure they can enlarge and extend the list in the near future. But talking about biostimulants, for sure we can consider plant extracts. So uh, biological and botanicals extraction can be used as biostimulants, along with seaweeds and protein hydrolysate and other compounds. So these biostimulants can affect the primary metabolism or the secondary metabolism, can have direct action by stimulating the plants, the different physiological pathway, or they can indirectly improve the crop productivity, like acting on the rhizosphere, for example, making some biostimulants increase the availability of some nutrients and directly can improve the plant's performance. But talking about plant biostimulants and derived from different botanical species, there are several examples, and there is also an extended research in order to find new botanicals that can be used as biostimulants. The most used is, for example, those that are belonging to the Fabaceae leguminosae family that are rich in protein that can be a source for production of protein hydrolysate, for example, Medicago sativa, alfalfa, or the leaf of Moringa oleifera that can be also used and is largely applied as biostimulants. But today I want to talk about borage, that is crop that is being used from a long time in, for example, in Chinese medicine and in food industry for providing different bioactive compounds that can have a positive effect and beneficial effect on the human health. So they could be also used for agriculture systems and used as biostimulants. This is part of research that have been done in the 4 or 5 years. And for characterizing biostimulants, as we said in the beginning, that the biostimulant has to act on plant productivity, have to stimulating the plants in using nutrients or increase tolerance against abiotic stress. So we can, for example, characterize the product just to see what they do on the plants. For example, looking the primary metabolism and see the extract from plants can enhance, for example, photosynthesis, increase chlorophyll concentration, increase or utilization of sugars, and so some macromolecule that can be used by the plants. In secondary metabolism, instead, is usually activated when plants can increase the tolerance against abiotic stress like drought, salinity, and so on. So they can reduce the accumulation of reactive oxygen species, or they can, for example, increase some antioxidant compound, or they can, for example, increase the water potential through the osmolytes accumulation and so on. So as part of our research focused on borage, we had a multidisciplinary approach in order to identify if the products can activate so different pathways related with higher growth or higher photosynthetic activity or increase the plant defense mechanism against abiotic stress. So this was applied in borage. So we used borage, in particular, water maceration, so water extraction. So we focused on the extraction of some water-soluble component, and they were applied in especially in lettuce and rocket. And I will explain why. Because lettuce is a good model system from agriculture crops for checking the speed of growth, and can be used for measuring the growth rate and the quality of the parameters related with leaf, and so with the chlorophyll or with the nitrogen metabolism. And also Diplotaxis tenuifolia, that is a rocket crop, is a good plant as a model for assessing the nitrogen use efficiency, especially nitrates, because we know that in leafy vegetables, for example, we have to lower as much as possible the leaf concentration. This for European regulations. And this can be also have some hormone effect. So since we know that from literature that borage has some different bioactive compound, we used two organs. First, the leaves and the flowers. Usually flowers are more concentrated in terms of bioactive compound, because it's an organ that must be defended by the plants. So all protection components are concentrated in open flowers in order to guarantee the fruit formation. So we are harvested the borage from field, and so we collect and separated the leaf and flowers, and we made the measuration for 21 days in within a ratio 1:2, in order to have a right concentration that can be in the same range of biostimulants. That means 1 or 10 milliliter per liter that will be applied in the crop. So we separated these two extract, and we applied in the crop. The first was lettuce, longifolia, so romaine lettuce. We applied two treatments during the cultivation, one in the middle of the growing cycle, another closer to the harvesting, in order to see the effect on the quality parameters. So we know that biostimulants must be effective in the low application. Otherwise, if we have to apply in line numbers, of course, the cost and the sustainability will be lower. So we have to find the right concentration in terms of doses and numbers of application to use. So what we found that effectively in 22 days during cultivation, especially the flower and 1 milliliter per liter concentration or 10 milliliters per liters, they had higher growth and the biomass accumulation, as well as 10 milliliter per liter of leaf extract, but 1 milliliter of leaf extract was not able to make a difference compared with the control. So in order to see if they have a biological function, we can see that we measured the proteins accumulation. Even when there is an activation of a metabolism, we have a higher number of proteins that can be translated in a plant. So we measured the total proteins, and we found that there is a trend in increase with higher concentration. The highest concentration was found in flowers in crop treated with the flower extracts and 10 milliliters per liters. Another important evaluation that was done is done photosynthesis activity, and we found that especially again those that showed the higher biomass accumulation was confirmed also by the photosynthetic net activity. You can see that it was higher in the treatment with flower extract 1 and to 10 milliliter per liters, and leaf extract in 10 milliliters per liters. Then we moved to the identification of secondary metabolism in order to see if the product can be also tested as a product to be used against abiotic stress. And we found that phenols, total phenols increased. It was significant, the increase, and was found in all treatments without any difference between flower or leaf extract and between 1 to 10 milliliters per liters. The same was found for flavonols, and the antioxidant capacity of the tissue that was measured was higher, but was not statistically significant from the control. But this can be also a positive effect for beneficial potential beneficial effect on the human health in the product that can be used by the consumer. In order to be sure that this activation was shown at the molecular level, we measured also the activity of the enzymes, the key enzymes of phenylpropanoid pathway, the phenylalanine ammonolase enzyme. And we found that there is an increase of the activities, and this was also confirmed by Western blotting using antibodies against the protein against the phenylalanine ammonolase protein. The second test was done in order to show if the treatment could increase the nitrate assimilation, so increase the nitrogen use efficiency. Was used the same scheme of application, but we use rocket because rocket is a hyperaccumulator of nitrates in leaves. So if the product has to lower the concentration of the nitrates inside the leaf. And we found also that there is an increase also in some sugars, sucrose, and total sugar, there is an increase, but was not significant from statistical point of view. As I said, nitrate concentration is a very important quality parameter for leafy vegetables and must be down below the several limits that can be differentiated between season, so winter and summer, and among crops, can be spinach, lettuce, or rocket, and in difference also when considered between open field and protected cultivation. This because the nitrate assimilation is related with light intensity. So lower light intensity, so it's more difficult to have an assimilation. And we know that from human health point of view, the high nitrates can induce some methemoglobinemia in children, can induce some gastrointestinal cancer, but can have also protective function in the stomach and as antimicrobial. But we don't want to discuss about the positive or negative effect of nitrates. We know that from a commercial point of view, we have to lower the concentration inside the leaves. So we applied the treatments in 10 milliliter per liter leaf extract and flower extract. We found that was almost halved the concentration of nitrates. And this was seen through the activation of nitrate reductase enzyme. We can see that there is a rapid increase in flower extract and lower, but also increased in leaf extract later after 4 hours of light exposure. So when we have to measure the nitrates accumulation, nitrates reductase activity, we have to consider the timing of light exposure of our crop. At the molecular level, we wanted to see, so the treatment affected the carbon on the nitrogen metabolism. So we look at different genes that are related with the uptake and assimilation of nitrogen. So it's a gene involved in nitrate reductase, glutamate synthase, and nitrate transporters. So, and those that are related with photosynthesis and assimilation, like rubisco, isocitrate dehydrogenase, and PEP carboxylase. And we found that effectively the treatment increased, and so these enzymes are circadian genes that are related with the time exposure and change during the day. So hour by hour, we can find a different expression, but we found that the application leaf extract, flower extract, they effectively induce the significant change in the transporter of nitrate or enzyme involved in the reduction assimilation of the nitrates inside the plants, as well as those that are related with the carbon formation, because the nitrate is reduced and attached in carbon skeleton to form amino acids. So it's important that we have enough sugars that can be used for nitrate assimilation. So in conclusion, what we can say that botanicals can from different plant species can be selected to produce biostimulant. They can be a good source of bioactive compound and for producing potential biostimulants. It's important that usually the biostimulants function depends on the homogeneous raw material. So we can use plant species, botanicals that we can grow in field or in greenhouses. We can make a standardization of the raw material. So we can produce anytime the same type of biostimulant with the same concentration of bioactive compound inside, without having so oscillation and variations and changes that can affect the final product. So this is can be really a good source for biostimulants. So the research has to explore the different botanicals that we can have, we can grow for providing a good source for the biostimulant industry. And this is more or less what I wanted to show you today. And we have several also publications related to this topic. So if you can go through our database, you can, or you can ask, we can send you the papers. Thank you very much for your attention.
Thank you so much for such an interesting presentation. This was a lot of information in such a short time. Let's give us also our audience some time to digest and come up with questions. So, Antonio, you mentioned essentially borage and using the flower and the leaf extracts from borage to deal with some abiotic stress. Can you detail a little bit more about the extraction methods that you have used? And namely, if you have categorized the water extracts and which components or compounds were related to some of the effects that you just show us?
Yeah, so we focused on the water-soluble components. So we used the leaf and flower extract that we made and mixed them. They ground them and they put in water, and we leave it to maturation for 3 weeks. This is the idea: is to get all the component that can be water soluble. That means that can be phenolic. Different phenolic. We have some degradation of proteins and formation of some carbohydrates derived and amino acids. This is more or less the concentration that we have. We can use maceration, that is the most simple, but we can use also, for example, some solvent like ethanol to make a faster extraction. But in that case, you have to remove all the ethanol, and then we have to resuspend the components in water. Or we can use also heat treatments. So we can use an extraction by heating the component, the material in water to 60 to 70 degrees in order to make a faster extraction in 1 or 2 days. If you use maceration, so is the native fungi and microorganisms that will make the extraction and the degradation of macromolecules that can be then transformed in short peptide and amino acids. The important issue is the sterilization, so the inactivation after the 3 weeks. Then in that case, you can use, so you can freezing the products, and then you can use later on some sterilization filter in order to remove all microorganisms and make stable your products.
Okay, thank you. That's also a comprehensive explanation. Hopefully, our audience will read the paper and can follow some of those procedures to give it a try, and then come back to you and share their results. That was great, Antonio. Thanks. We have a question here from Pedro. So regarding the nitrates foliar concentration levels that can be risky, do you have the tables that indicate which
Yes, I
values we are
showed the
talking?
table. I showed the table that so that I can show again. So for by law, we have to keep this concentration in our products. So the problem is that we know that from human diet we have a different source of nitrates. Can be water, and we can have the nitrate also as a meat preservative, like in nitrate, sodium nitrate, potassium nitrate, and so on. We cannot remove the nitrates from our diet, so we can reduce the amount that we usually can intake day by day. And one is especially to reduce the source from vegetables, and especially in leafy vegetables that can have a high amount of nitrates. This is the regulation and the limits for different crop in a different season and in different growing systems, greenhouses or open field. So for spinach can be 2,500 for example in summer, and maximum 3,500 in winter, and so in different concentration also in for different season. In lettuce maximum 5,000 or 4,000. For example, rocket salad, so sativa, Plotaxis can the limits are higher because they are an hyper accumulation. So 7,000 in winter and 6,000 in summer.
Okay, thank you. That's a good indication. Alexander, just conscious of time, I will now move on to our second speaker. If we have time at the end, I have here a personal question, not personal to you, but I am curious about something that you raised. But let's leave this for later on if we have time, because I would now also like to give the floor to Loredana. Hello, Loredana. I know you also have something very interesting to present to us. There's something quite novel, right, Loredana?
Yes, exactly. Okay, I will share my screen. Sorry, but maybe. Okay, can you see my screen?
Yes.
Perfect.
Not in presentation mode, but
yes, I will move. Okay. Good afternoon to everyone, and thank you very much to you for the invitation. I'm very happy to share with you the knowledge about this very innovative molecular tool for tracking microbial inoculant. So this means species specific microbial species, target microbial species in soils for supporting also their application in soil. Just a brief overview of the context where we are. We up to now have focused on biostimulants. Now we focus on the beneficial microorganisms that are used in agriculture and compose microbial-based bioproducts. So named also biofertilizer, again biostimulant or biopesticide, depending also on their function and their application to support plant nutrition and protection from abiotic and biotic stress. microbial-based bioproduct is receiving continuous and increasing attention, considerable attention in the last decades by manufacturers, farmers, researchers, because they help to reduce the use of chemical in agriculture. So we can trust in and we can imagine a world without chemical input, but we have still to face several problems raised by the use and application of this product in agriculture. And even if they are currently widely used, statistics reports that we have around 10 billions for biopesticide and 3 billions for biostimulants sales around the world. We still have some problems related to the effect, to the impact that they can have once they have been applied in the soils. Problems are related to the changes that can occur to the soil microbial structure that can, of course, consequently affect the overall soil health status, impacting crop productivity, quality, and human health. But we know that at the same time, the efficiency and the efficacy of the microbial-based product may be affected by soil chemical and physical characteristics and by pedoclimatic conditions. So, how to say, it's challenging, it's a bit complex, considering also the very high complexity of soil ecosystems, to understand which are the drivers that can affect and that can shape the soil microbial structure independently from the application of those products. So some time ago, around 10-15 years ago, we started to ask us how we can solve and cope this problem. Also developing a new tool for monitoring the presence and the persistence of this bioinoculant, so of target microbial species used in those products, to understand, to follow their persistence in the soils. And this means that the field application of this product require their registration at European national level, together also with an indication by manufacturer of various specifications. And within this specification, the manufacturers needs to include also method for tracking and to follow their persistence and their fate in the soils, because they are living organisms, are not chemical, and being living organism, their destiny in the medium or long term timing can also be important to understand which are the effect and also the legacy that they can have on the soil microbial community and in general on the soil health. So the tool, a tool that allows us to follow, to monitor the persistence and the fate of this microbial inoculant in soil is considered important for farmers, also to confirm their successful inoculation and their persistence in soil. Is important for regulatory purposes, because we saw before that manufacturer has to include in the registration dossier a method for tracking microbial inoculant persistence in soils. But it's also important to evaluate the effect on native soil microbial community and detect them once they have been applied. And this is a kind of information that can also support farmers in the application of this product in terms of dose, but also of timing. Sometimes we listen that bioinoculant didn't work, but this it's not doesn't not necessarily means that certain bioinoculants are not have not been efficient and have not high efficacy. Sometimes it's a problem related to their establishment in the soil, considering that they have to compete with native soil microbial community. So the mechanism underlying the fate and the persistence of bioinoculant in soil can be the results of a sum of multiple variables and therefore be difficult to understand and predict. The introduction of any microbial inoculant to the soils from an ecological point of view should be also considered that as a disturbance to native microbiota. And the changes that we can observe after the application of this product could also be due to direct effect or indirect effect, for instance, because of nutrient released from dead inoculant cell or the inoculant root. And this is another reason why it's important to monitor the persistence and fate across the time of these species, inoculative species in the soil. Also, the survival and thus the efficacy of microbial inoculant in soil can depend on the soil native microbiota, and the effect might change over time and also in the space. Most studies, for example, measure the effect only over a short period after inoculation, but without following the persistence and phase. So just they did an impact evaluation of the effect without knowing effectively is 2 months after the inoculation, this product are still persistent in soils. So the method applied can differently represent the impact of bioinoculant, but sometimes are not informative effectively on the establishment and persistence in the time of this product. And this is reason why it's important that we not only look at impact evaluation, but we look also at monitoring of the persistence and the fate in the soils. Which method can we use to develop a tool for tracking a bioinoculant in soil? For several years and still now, we usually use a DNA-based method, and we exploit molecular or functional markers in order to detect and to have to design a species-specific detection of these species in soil. But we have to rely to the genetic variability of the individual strains, and this is this poses some problem and make highly challenging this kind of work. Because we have to face with the bias and with also the data that are located on BLAST, on NCBI. So we have to rely to the genome that we know up to now. And we know that we don't know all microorganisms occurring in the soils. And when we design a species-specific marker for the traceability of target microbial species in the soils, we have also to mind that probably we are designing such a marker, such a probe that can also recognize another microorganism still unknown that occur in the native soil microbial community. And this is one of the things that make too much difficult our work in designing a species-specific marker for the detection of a bioinoculant in soils. But usually, we start from the genetic characterization of the bacteria or on the fungi that we have to detect, to track, in order to throw a pangenomic analysis, so the comparison of all the genome that are closely related to the genome of our target species, in order to identify the unique trait, so the genetic trait that can effectively distinguish, can allow us a discriminant detection of our species in the soils. And usually, we also start from the whole genome of the microbial species that we have in our hand, in order to design a highly specific marker for the detection of a target species. And we then use a PCR-based method for tracking, for measuring their presence in the soils, both presence and quantity, sometimes using real-time PCR. But all the PCR-based method has showed pros and cons, and sometimes they are too much expensive, are highly time demanding. They request a proper soil sample collection, and this sometimes represent also a further problem for us, because we need to collect fresh soils and to store immediately the soils that we have to process for downstream analysis, in order to not affect the results. Considering the bias, but also the processing time, that we can have during the preparation of the samples. This is a table that some time ago, in a review that we have published, to present an overview also on the literature about the application of this product, and the study that has been carried out up to now to detect and monitor the impact, evaluate the impact and monitor the presence of bioinoculant soil have been done. And we effectively summarized there the advantages and limitations per each technique that have been widely used to do this kind of work. And also the needs that we have for each technique. And we realized that probably also to meet the needs of the farmer, we shall, how to say, shall look at to effectively meet the needs of the farmer. We need to develop a non-field tool, a non-real-time tool, in order to effectively provide to the farmer a tool to monitor the presence of this microbial inoculant in soil, and to effectively exploit the results related to the presence and the persistence of microbial inoculant in soil once applied, to optimize their application in terms of timing and dose. And within Excalibur project, that is an European project that is coming to the end, we developed a new and innovative tool to track and monitor the presence of microbial inoculant in soil after their application, always using a DNA-based technique for binding species-specific gene sequences, but not marker, primer, or probe that we can use in a PCR-based approach, but we started to look at aptamer. What are aptamer? Aptamer, as showed in this table, are comparable to antibody, and they works out as antibody. That means that they can specifically recognize a bind, a target, but without recognizing at cellular level the organism. So it's not necessary to extract the DNA. And this represents the first very big innovation, how to say, important, significant innovation, because this means that we don't need to collect soil and extract DNA. We need to collect soil and extract cell. And the cell extraction from the soil is not expensive. So cost has a very low cost in terms of equipment and material that we need to use. Is a quick protocol that we can apply in few minutes, in an half of hour. And so you can imagine how these can represent, how to what does extent this innovation effectively can revolutionize, can change effectively the diagnostic of this product in the soil, in a complex matrix like soils. So we look at aptamers also because they can be immobilized, they can be modified, attaching, for example, a fluorophore, so they can be detected also through fluorimetric approaches. They are stable. They can be they are more stable also in higher temperature. They are active in different reaction environment. So compared to the antibodies, they show a lot of advantages that make us very fascinating and promising for diagnostic applications, also in the fields of soils, because up to now the literature is very rich and abundant in showing several applications of aptamer in the fields of food contamination diagnostic. So they are aptamer in se is not a new technique, but the innovation is related to their application in the soil. And it's related to the fact that we, after 2 years of hard work, we effectively were successful in designing an aptamer specific for the detection of Bacillus subtilis in soils. Why we started from Bacillus subtilis? Because Bacillus subtilis is a plant promoting plant growth promoting rhizobacteria, is widely used in some biofertilizer and biopesticide, also in some bio-based product, and is also widespread in the soils. And this also allows us to have how to say, a good scenario from a technical point of view to design an aptamer species specific for Bacillus subtilis. And monitoring and tracking Bacillus subtilis in soil is again key to optimizing not only the binoculum application in terms of dose and timing, but also for effectively understand the effect and on soil microbial community and the efficacy and efficiency of this product once it's have been applied. And considering that aptamers are emerging as a biosensor based on single-strand DNA or an RNA capture probe that can effectively bind various targets with high affinity, and considering that they are cheaper, they are more sensitive than antibodies, they can be mobilized, and they can also be cost effective. We decide also to transfer all the knowledge, so immobilize this aptamer on a portable device. But before to show you briefly which is the device that we are building, I want to briefly show you which is the work that we did. Here I linked, I included the paper that you can read if you want to go in deeper detail. But briefly, we started from this species of Bacillus subtilis, that is a particular strain included in a commercial already marketed product available for agricultural application.
Loredana, just to give you a note that we are slightly over time, okay?
Okay.
Thank you.
And thank you, Claudia. And we compared around 500 different genomes to select the unique trait on which we have finally designed the library to identify the aptamer candidate. After the selection and the validation of two different aptamer, one with strong affinity for our strain and the second one for strong affinity for Bacillus subtilis strain, generally speaking, we decide to use a quartz crystal microbalance to transfer, to immobilize this aptamer in order to develop a biosensor to be used in on-field application for a very rapid, precise, and low-cost monitoring on field of the bacterial concentration in soils. So, in conclusion, the chance of designing custom and specific probe with high specificity increase with the genetic characterization of the species involved in the formulation. And this was what we did in a Excalibur project on several species, with a particular attention on this species and on a fungi on which we have designed an aptamer as probe for their detection. And the application of classical molecular technique should be used in designing a proper and specific tool. And the lab-on-chip technology, as we are testing on field, can effectively open the avenue for the parallel high throughput multiple identification of target microorganisms and genes in environmental samples, as we are indeed working on combining different aptamer in order to have multiple identification of different microorganisms, considering that on the market we don't have a single inoculant, but we have sometimes products that are made of consortia. So it's important for us, the multiple recognitions, and the aptamer allow us to do that. We are aware that the traceability of target microbe in soil is challenging, but we think that a robust knowledge, genetic, functional, metabolic knowledge of the microorganism can pave the way for an easy and robust identification of a specific probe for the detection of microbial inoculant in soils. And despite all the challenges posed by the soil complex matrix, we think that all the effort put in place, in particular in Excalibur and now in SpinFert project, for develop the monitoring microbial inoculants in soil can effectively boost and support the adoption of this product for agriculture. So that's all.
Thank you.
Thank you.
Thank you so much, Loredana. Definitely, you've addressed a very important topic to understand what species are still there and how our biostimulant and our inoculant working. So I'm sure this is a very promising tool that we are very eager to see in the market. Loredana, I know that you have explained this, but could you just detail again and summarize and maybe emphasize the most relevant points on how aptometers achieve target recognition at a cellular level without requiring nucleic acid extraction, and how these methods of bypassing, these extraction steps relates to some cost savings compared to the more traditional methods?
The aptamer can assume a secondary or a ternary structure. And these make them comparable to antibody. Because as I showed before in the figure, effectively, I want to show you again so it's better. Oh, okay. I don't know how.
We can see your presentation.
Can you see now again my screen? Okay.
Yes.
You see there, this is an example of the aptamer structure. Okay. They exploit they are able to ligate the surface of the cell. Okay. But the process through the protocol, the methods through they have been selected, allow them to effectively recognize only exclusively the cells for which they have been selected. Because I didn't show you the protocol because it's very specific and in my opinion, ask for a dedicated presentation, okay? Because it's also complex to show. But briefly, you design a couple of primers on your unique trait that you have identified through the pangenomic analysis. I mean, through the comparison of several genomes closely related to your species. You identify the core genome, that common and unique trait. On this unique trait, you design the primer and you build a single strand library. And you use this single strand library through 4-5 rounds of a PCR against your species continuously in order to select the sequence of candidate aptamer that effectively is specific in recognize the cell of your target species. Okay. We are now also working by using the electronic microscope to effectively see where, in which part of the cell aptamer ligate to recognize and also the force of the ligation depend from the chemical characteristic of the aptamer. Indeed, we have selected this aptamer within 6 candidate aptamer with different chemical characteristics, okay, binding affinity and so on. And this is reason why you don't need to extract the DNA, but only the cell.
Okay. Interesting explanation, Loredana. I think we could stay here for a very long time, and there's a couple of questions that
It's
this
a
raises. It's very
fascinating.
interesting. Definitely.
I know it's a fascinating
Yeah,
protocol, but it's also not easy to.
Yes, it's very new. I realize
Yes,
that. So it's,
to understand.
thank you so much.
Yes.
We now need to
Thank
really
you.
need to
Thank
move
you very
on. We
much
have
for
Alexander
attention.
and thank you, Loredana. And he has a new star to show us, Leafamine, right, Alexander? So Alexander, thanks for sharing. And so let's hear what Leafamine can do to water stress tolerance.
Good afternoon, everyone. My name is Alexandre Kerckhove. I'm a PhD student at Institut Agro Rennes, and I work with both BCF Life Sciences and the Institute of Research in Horticulture and Seed in France. And today I'm going to present to you some of my work as a PhD student, which will be on Leafamine, which is a protein hydrolysate biostimulant, and how it enhances lettuce tolerance to water stress. So first of all, you have to know that drought stress is a huge threat to worldwide food security, because as you can see on this graph, the world population goes up very fast. In 2050, it's predicted to be around 10 billion people. But on the C graph, you have the billion of hectares of crop production land, and you can see that it doesn't grow almost at all. And the black arrow shows what we would need in billion of hectares to feed everyone. So as you can see, we have a big lack of land. And if we look at the billion of cubic meters of water we would need to irrigate all this crop production, we will have to irrigate with 4 billion cubic meters. But the black arrow shows how many we will have available to irrigate. So as you can see, we will lack a lot of water and a lot of land. So we have to both improve yields and use less water if we want to feed everyone by 2050. And in France only, climate change causes an annual loss of 1.2 billion euros, and drought is responsible for 60% of that only. So what is drought stress? Because it's a small word, but it says it's a lot of things. It's a cascade of morphological, physiological, and biomolecular responses. Depending on the intensity of the stress, you will have different response. And the stronger the stress, the harder it is for the plant to respond, to have a response to it and to grow. For the morphological response, you have things like reduction in biomass and a poor floral development. The biochemical and molecular responses are the reduced water accumulation and an increased abscisic acid, a decrease in efficiency of Rubisco and everything. And for the physiological response, you have things like a reduced stomatal conductance and a reduced chlorophyll content. And all this leads to the response of the plant, which is a reduction of crop quality, a stunt growth, a bigger senescence, and premature cell death. And when I say that I want to enhance plant tolerance to drought stress, what does it mean? So when a plant suffers from drought stress, it's going to be smaller and to suffer from different sickness. And enhancing plant tolerance to drought stress means that you want, with the same amount of water, to have a bigger plant with a better quality. And how do we do this? We need to maintain photosynthesis. We need to have more antioxidants. We need to have higher osmotic adjustments, maintain the stomatal conductance. We also need to stimulate the hormones related to drought stress tolerance, like abscisic acid. We need higher root growth and a bigger leaf area. And all of this leads to a maintenance of yield. So for our plant model, I work on lettuce. Lettuce goes from the seedling stage to the commercial stage in 70 days. So it grows really fast. It's a worldwide production. The world lettuce market is $3.5 billion. And it's highly sensitive to water deficit due to its shallow root system. The roots doesn't go deep in the soil. And it has a high water content, around 90%. And so yield in lettuce is very affected by water deficit. As you can see on this graph, a land which is correctly fertilized produces around 3 kilograms per square meter of lettuce. And when you apply water deficit, you lose almost 30% of it. And biostimulants were said to be a solution to that because they can significantly enhance lettuce yield under both optimal and water-limited conditions. And in this study, when they applied a biostimulant, they got back almost half a kilogram per square meter, while still being in water deficit. First thing you need to know about biostimulants is that they are classified into 6 families. First one being humic and fulvic acids, protein hydrolysate and amino acids, seaweed extracts, microbial inoculants, inorganic substances, and oligosaccharides and metabolites-based biostimulant. But today, I will only speak about protein hydrolysate and amino acids because that's the product I work on. So, a small recap on protein hydrolysate is that a protein hydrolysate mostly come from waste from the agro industry. You can make protein hydrolysate from plants, from blood, from chicken fevers, and you get your protein source, and you can hydrolyze it either by chemical hydrolysis, which will give you a lot of free amino acids, or by enzymatic hydrolysis, which will give you a lot of peptides. So depending on your strategy and your protein source, you will have a different mix. But a protein hydrolysate is a mix of free amino acids and peptides. And this family has shown a lot of effects on plants. They have a good effect on flowering and fructification, photosynthesis, osmotic adjustment, growth metabolism, root architecture, and a lot of things. And overall, they provide a better yield, a better plant growth, and a mitigation of abiotic stress responses. So, about the product I use, it's called Leafamine. It's made by BCF Life Sciences, and it's a product of circular economy. So BCF gets the chicken fevers from the poultry industry in Brittany, in France, and to recycle it, because it's a waste, so it was going to be destroyed, but they get it, and they make amino acids out of the keratin of the feather. This is the composition of Leafamine. It's a mix of 17 amino acids, with the most abundant being proline, serine, and glutamic acid. So Leafamine is 88% of amino acids, 6% of peptide, mostly di and tripeptide, 82% of free amino acids. All of them are L form, which is the only form that the plant can absorb and use. It has a low molecular weight, it's stable, microorganism and heavy metal free, and you can use it in organic farming. So about the experimental protocol, we use, as I said, lettuce. In long day condition, we have 2 conditions of treatment, with or without Leafamine at 5.57 grams per liter. We do 4 treatments, 1 per week, and then we sample with a lot of things for our lettuce, and we have 2 water conditions, well watered at 100% of field capacity, and a small water deficit, a mild water stress, at 70% field capacity. So first thing is you can see is that in well water condition on the left, this is the difference we have on the photograph of our lettuce. On the left, the control plants, and on the right, the Leafamine treated plants. As you can see, Leafamine treated plants are bigger and greener. And when we quantify the average biomass, it goes up by 33% in well water condition. And for the water deficient, you can see the same thing. We have bigger and greener lettuce, and the average biomass goes up by 20%. When we look at the number of leaf and leaf area, the number of leaf is in full line, and the leaf area is in dotted line. You can see that for the number of leaf, it doesn't really change anything applying Leafamine. But on the leaf area, we have on well water plants a 25% rise. And for the deficit irrigated plant, we have the same rise of 25%. Then we wanted to know what happens. Why do we have bigger lettuce? First thing we have is that we have a bigger chlorophyll content. On well water plants, the chlorophyll content goes up by 43%. And on water deficit lettuce, it goes up by 19%. Another part of photosynthesis is the stomatal conductance. And when we quantify stomatal conductance on well water plants, there is no difference between control plants and Leafamine treated plants. That means the stomata are both open. But when we apply a water deficit, you can see that we lose almost half of the stomatal conductance, which means our stomata are mostly closed. And when we put Leafamine, as you can see, we goes back up to the level of the well water plants. So it means that the stomata are back open, with an 88% rise in stomatal conductance. And for the photosynthetic rate, which is the quantification of CO2 absorption by the plants, as I said, we don't have any change for the well water plants. But when we look at stressed plants, we have a photosynthetic rate that goes up by 62%, which means we have way more photosynthesis in stressed plants treated with Leafamine. Then we're going to abscisic acid concentration. So abscisic acid, as I said earlier, is the phytohormone related to water stress. It's produced during water stress, and it's a hormone that is going to make the plant's responses to everything about water stress. So it's normal to not have any in well water plants because the plant doesn't need it. But when we go to the water stress plants, the quantity of ABA goes up to by 70%, which is a lot. And another thing Leafamine does on this lettuce is that it improves osmotic adjustment and antioxidant activity. So on the well water plants, we can see that we have soluble sugars that are overexpressed, and we have amino acids like lysine and tyrosine. And for the water stress plant, we also have soluble sugar, ribose, galactose, and we have a lot of amino acids. But why is it useful to the plant? It's because amino acids are what we call osmolytes, which helps the plant to maintain its water content. And some amino acids like proline has a double effect. Proline is either an osmolyte, a well-known one, but it's also an antioxidant. So they both have this double effect, and it's both very important for the plant if it wants to grow. So if we come back to this graph, we have already shown all of this with Leafamine, but we still have one thing we need to show, is that we need to see what happens in the root system. And that's what we're going to see now. But first, why do we want to study the root system? It's because root architecture provides an in-depth understanding of plant response to biostimulant application. The roots are the place of mechanical anchoring for the plants, mineral and water absorption. There is a constant interaction between the root system and the above ground parts, with nutrient exchanges, hormonal signaling. It's also the zone of symbiosis and mutualism for PGPR and nodules. And it's also the place of environmental perception, biotic or abiotic. And how do we study the root system? We use a novel Rhizobox system that we made. First of all, you have a plexiglass frame with rubber edges to all the substrates. You put substrates inside of these rubber edges. After that, you close the Rhizobox by a piece of fabric and a plexiglass sheet. The plant goes between the piece of fabric and the plexiglass sheet, and the whole point is that the plant never goes inside of the substrate, so you can see the root grow, and you can sample roots without destroying the plant and without doing anything to the plant. You just have to open the box, cut the roots, get it, and close it back. And in the end, we put a lid on the roots to protect it from the light. And that's what it looks like when it's been used in real time. What we showed on Rhizobox at this time is only on well water plants. I'm not going to talk about water stress plants currently. What we saw on well water plants is that, as you can see on this picture, we have a root system that seems to be the same, but it also seems to be thinner if you look closely. And when you look at the aerial part, there is no doubt the Leafamine treated plants have a way bigger leaf area. So when we quantify this, what we see is that the leaf area of Leafamine treated plant goes up by 36%, and the roots area goes up by 33%, which means we both have a bigger leaf area and a bigger root area. When we look at the fresh and dry rates of leaf, we can see that the leaf are 53% bigger when we apply Leafamine, and the dry weight follows by 15%. And for the roots, the root weight is going down by 45% for the fresh weight and 44% for the dry weight. So now we are in front of a problem because we have a bigger root area, but we have less roots when we look at the weight. So how is it possible? And when we want to look at the density, which means the thickness, we can see that leaf thickness goes up, but the root thickness goes down by a lot, minus 60%, which means that the plants produced more roots because we have a bigger root area, but thinner ones. So it means that it needs less resources to grow. And when we look at the shoot-root ratio of Leafamine treated plants, it goes up by 44%. And the shoot-root ratio is an indicator that says whether the plant is privileging the higher parts of the root system. And if it goes up, it means the plants prefer the higher parts, the growth of the higher parts, over the growth of the root system. So to conclude on this work, what we showed in well water plants is that when we use Leafamine technology, we have a bigger plants with more roots, but thinner ones. How do we explain that? We cannot explain this by photosynthesis because, as I said, the photosynthesis is globally the same. But when we look at the sugar flow, by looking at the root ratio and its growth, we can see that if this is considered as a normal sugar flow for the plants, when you use Leafamine technology, the sugar flow, which goes from the leaf to the roots, is reduced. We have 60% less weight because as the root system is already bigger, with thinner roots, it covers a bigger area. The plant doesn't need more resources for the root system, so it provides all the resources for the higher parts and the growth of leaves. And when we look at the stress plants, using Leafamine technology provides a bigger lettuce, greener, that looks healthier. And how do we explain that? As I said, the stomata seem closed on the water stress lettuce, and using Leafamine puts the stomata back open, which leads to more photosynthesis and a bigger plant. We don't know yet what happens in the root system. That's what I'm trying to show currently. And what we want to do now is to complete our understanding of the effect of the biostimulant on root architecture and growth kinetics under both well-watered and water-deficient plants. We want to identify what happened inside of the plants with the early physiological and molecular mechanism behind the regulation of root and shoot growth. And all of this is going to be useful for us to provide a comprehensive picture of the holistic effect of the biostimulant at the whole plant level. I just wanted to thank my team at both PCF Life Sciences and the Institute of Research on Horticulture and Seed. And I'm going to just invite you to read our publication of this topic. And thank you for your attention. I'm going to answer every question you will have. And that's it.
Thank you so much, Alexandre. We can tell that you are passionate about your PhD topic, so that's very rewarding to hear and very interesting work that you have presented here. So we've heard about the Leafamine effects on water stress lecture. Can you summarize for us what are the key advantages and also discuss how these benefits that you identify compare or eventually complement some other drought tolerance solution that we already have in the market?
Yes, so I'm just going back to this one. So first, Leafamine is very interesting about how it's produced. As I said, all or mostly all of the protein hydrolysate biostimulants are made from waste. So first, it's a way to recycle all the waste from the agro industry. And it's completely biodegradable. So when you put a biostimulant on the plant, it's going away, like it's not staying in the soil, everything. And it provides help for the plant at all levels, because we have also seen that putting a protein hydrolysate biostimulant, not only Leafamine, I'm going to speak about the whole family here, because most protein hydrolysate biostimulants have a big part of their effect in common. And using protein hydrolysate biostimulant also enhance the development of microbial community. We have more microbes when we use protein hydrolysate biostimulant. And we also have more exchanges between the plants and the microbial community, because as you may know, amino acids are like the second most molecule that is the most traded between the plants and the microbes during symbiosis. And overall, actually, to protect the plant from water stress, we didn't really have many solutions. Some people used drip irrigation, which is like to irrigate only what the plant can absorb slowly, but you need a lot of technical things, like you need a whole system to use this. You also have like some biopolymers that you can put in the soil and that are going to retain water. And apart from that, we didn't really have a lot of things to protect plants from water stress. And biostimulants are able to help the plant grow, either on the well water plant, because you can use it on plant that doesn't suffer from any stress and is going to be better anyway, because it helps to absorb the nutrients. It helps to a lot of things, actually, for the hydrolysate. I'm not going to say it all again. But
and Alexandre, do you think these results can be transferable to other crop types? So you mentioned lettuce, very focused on lettuce.
Yes.
Can you give us an idea if this would work in different crops?
So I won't cite it all, but the company I work for, BCF Life Sciences, has tried Leafamine on 170 crops, different crops, almost all of the crops you can think of, corn, tomato, strawberry, every big crops. And it has a positive effect on all of them, which is very different between the plant. I mean, on lettuce, you have a bigger leaf area. On tomato, you have more fruits. So it's not the same effect on every crop. But on all of these crops, we have a positive effect, which can vary a lot. I mean, even of two varieties of lettuce, you can have different effects. I mean, on one variety, you can enhance the productivity by 30%, and the other one by 20%. But on all of these crops, we add a positive effect. So yes, biostimulant can be used on mostly all crops you can think of.
Okay, good, interesting. So, but unfortunately, we are running out of our time. We have a question in the chat, Alexandre. I kindly invite you to answer in the chat. I apologize for our audience. We don't have more time for questions, but please tell us if you want to know more and get in contact with our speakers. I know that some of you are eager to start answering our quiz. So we have prepared a couple of questions based on the topics that we've heard here today. Some very interesting presentations. We heard about borage and its relationship to abiotic stress. We also heard about aptometers, so how we can detect microbials in a more cost-effective way. And lastly, Leafamine with Alexandre, also promising avenue to deal with water stress. Thank you so much. So if you are interested, please join us here at this link in the forms, fill in the questions, and we'll make sure that we can send you a certificate from your participation here in this webinar. So for me, it's not all, folks, but we're going to have a pause for a week because we come to the end of this webinar cycles. We have addressed the biostimulants, and I think we have covered quite a lot. So there's a lot of food for thought for you. But on the 3rd of April, a Thursday, at the same time, so 4 p.m. Portugal or Lisbon time, we will start addressing the topic how we can transition from pesticides to biopesticides. And we have cooked for you some very interesting sessions also on this topic. So I hope you can join us later on. In the meanwhile, if you have more questions about the webinars, about our speakers, please get in touch, and we are here to assist you. It was a pleasure to be here discussing more about biostimulants, and I hope that we can see you all in the next session, okay? And I also take this moment to thank our team for making this possible. And I can see here Rita and Soraya, thank you very much. Also to your support to make these sessions possible. It's always a pleasure, and things always run very smoothly when you are around. So please, once again, thank you. Enjoy your evening. And to our speakers, also many thanks for your presentations, for sharing your expertise, and hopefully we can be in touch for more projects and collaborative approaches among our institutions. Okay, so nice to see you. Hope to see you in 2 weeks' time. Bye.
Thank you very much. Bye.
Bye.
Thank you, everyone.